we do have, we think we might have some dinosaur collagen. That's actually being highly debated, though. But the collagen, I mean, it's just one protein. You couldn't rebuild an organism knowing one protein. But some people have speculated that you could look at, the closest living relatives of dinosaurs are birds and crocodiles, and so you could use this process to try and reconstruct a dinosaur genome. But that's to have the whole thing work would be a genome is so big,
awesome,
and there's so many different molecules involved in there. Plus, we don't have the eggs,
exactly.
And you need it's complicated. But we could definitely resurrect extinct proteins that way. Not extinct animals, but extinct proteins. We can, yeah.
Dang. There was one more question too. Someone asked, do punctuated equilibrium compete with gradualism?
Punctuated equilibrium has more to do with phenotype, and the evolution of molecules happens slightly different than the evolution of phenotype, and I can explain that a little bit more. Maybe just explain that right now. What's discovered here is not at odds with punctuated equilibrium. Punctuated equilibrium is this thing that we find in the fossil record, that we see fairly rapid changes in form of animals, and then we see long periods where they don't change in form at all. During that time, mutations are adding up in their bodies. All sorts of mutations are adding up. But when a population is large and everyone's interbreeding with each other, those mutations get spread out across the population in a way that you can't really see them. You can't notice them on the large scale. You can't see it in their skeletons or whatever. When a major selection event happens, like climate change event, a new predator or something, and the population gets whittled down, all of the best traits that already exist in that population that are hidden because they're interbred into the entire population, all of those best traits get manifest all at once in the tiny population that survives that catastrophe. And that's where we see major evolutionary change. So the change is happening constantly, but we only see it when there's a bottleneck in the population. That answer the question?
I think so.
Okay. Let's do next slide here. Oh, wait, actually, let's stay on this one for a second. Sorry.
Poor
mutations
Dave.
happen at a somewhat... I'm trying to read my own writing here. It's hard online here. If mutations happen at a somewhat regular pace, each mutation represents a roughly equal unit of time. We just don't know what that time is. Linus Pauling speculated that it was several millions of years for hemoglobin. And it's that speculation that Nathaniel Jeanson, of course, would disagree with. But Nathaniel does agree that each mutation represents some period of time. Yeah, because these mutations happen at a roughly regular pace. It's not as perfect as the ticking of a clock, but it's sort of there. And the next slide, I talk about this. How do you get regularity out of randomness? It's a really weird thing to think. But if you look at our DNA, we've got 4 letters that we can work with. We've got 4 different types of nucleotides. So that's essentially like rolling a 4-sided die, right? You've only got 4 options. Every time a mutation happens, there's only one of 4 things are going to happen, right? And if you roll a dice, if you roll a normal 6-sided die and you roll it you roll it 6 times a minute, you're going to roll a 6 once a minute. So you get regularity from a random process, right? Now, there's a problem with evolution, though, and that is that not only can you just roll the dice, not only can you just mutate individual nucleotides, but you can also add extra nucleotides, you can take nucleotides away. All sorts of weird things happen across evolutionary time. And that's one of the reasons why these clocks aren't perfect. That's why there's those jagged edges that we saw on Nathaniel's tree that he had made. I don't know if he made that tree, but it was in his book. And the other thing that makes evolution not so regular is that we're not necessarily rolling the dice every 1,000 years. Maybe we roll the dice every 100 years for a while, and then we only roll the dice once every 10,000 years for a while. So the time changes, and we actually know several things that cause the rolling of the dice, the frequency of the rolling of the dice to change. So there's a lot of things that people have been studying and trying to figure out that influence the speed of molecular mutation, and that might affect it and might be screwing up our evolutionary clocks, our molecular clocks. And this is a very active field. It's called coalescent theory. And people who work in coalescent theory, like Dr. Mays, they are using statistics. They're combining statistics with observations. They're learning everything they can about how genomes mutate, what types of things affect their mutation rates, so that we can add those into these equations so that we can make our evolutionary trees more and more accurate. We would love not to only have the relative time in which different species diverged. We'd love to have the actual time. We'd love to be able to know in years how long ago two things split. We can't do that right now. We can't do that very accurately right now, at least. We still depend on dating fossils to figure out when things actually happened. And then we use those dates to calibrate, for the most part, we use those dates to calibrate these molecular clocks. And that is something that Nathaniel Jeanson doesn't like, obviously, because we're using this millions of years data. And you can make the argument that this is circular reasoning, right? We're using the radiometric dating to date fossils, and we use that data to calibrate the evolutionary clock that we get from molecules. And that is a type of circular reasoning, but what it does is it allows us to, we've studied very deeply the separation in the fossil record between the human lineage and the chimp lineage, and we're pretty confident when that happened just based on the fossils. There's a whole bunch of other things in human history that we are not confident about, and so we can calibrate to the human-chimp split, and then with that calibration we can use molecular data to figure out the dates of other events in our own evolutionary history and even more recent history, like when different areas of the world were colonized by modern humans. And we can use that to give us information on where we should look for fossils and where we should look for archaeological sites. And that's something that we're now doing. We're using that to, it's a very new concept, but we're now starting to use this to try and help us find fossils. And Mays would be able to tell you a lot more on, if we've had any success doing that or not. But that is what we're now trying to do with these clocks. And you have to get a year. Yeah.
Sorry. So it could have some predictive validity then. You could formulate a hypothesis based on the corroboration of these molecular clocks, and also our understanding of the fossil record and our use of radiometric dating. If you can corroborate all of those together and conglomerate them, then you can make a testable hypothesis, kind of like what was the fossil that they predicted that we found? Tiktaalik, yes, kind of like Tiktaalik. So you can first, to a certain degree, obviously, you can still make predictable hypotheses based on the consolidation of all of this information as well.
It's consilience of everything, right?
Right.
Yeah.
A cooperation.
That all depends. That all depends on us being able to figure out the variables involved in here. What's dictating the rate of the clock over time? Does the clock change? How does it change? And I'm going to show you in a little bit a calibrated clock that's working super well. I mean, it matches up with all kinds of things that we find in the fossil record, and it's surprisingly simple, the math that's involved in it. So yeah, let's do the next slide here.
Next slide.
So what types of molecules might be best for making these clocks? And the answer there is, we already know that it takes 3 nucleotides to code for 1 amino acid. So that means that proteins, they're pretty cool, but they're not as good as DNA. So DNA is the better molecule to use if you're going to make an evolutionary clock. It's got higher resolution, right? And then the question is, do you want to get that DNA from the nucleus, or do you want to get that DNA from the mitochondria? And maybe you want to use both. Let's go to the next slide here. Here I'm just talking more about why DNA is better than amino acids. Is that concept clear, or should I explain? There's actually a really cool example with dolphins and bats, where if you use amino acids, you get the wrong evolutionary tree. If you use DNA, you get the right evolutionary tree. Should I explain how that worked?
Sure.
Yeah. So that this there is a protein that all mammals have in their ear called prestin, and it helps fine tune the types of sounds that you can hear. And so mutations in it will cause you to hear sounds that are lower, and other mutations will cause you to hear sounds that are higher. There's a type of, I believe it was a porpoise. It might have been the bottlenose dolphin, but I think it was a species of porpoise that has the exact, in certain regions of the gene, it's more similar to a bat's prestin gene than it is to any other whale's prestin gene. And it turns out that these two animals, using echolocation, have hyper-focused their attention on this one frequency of sound, this very specific frequency of sound that works really good for echolocation. And that's why they've got these same amino acids, the same amino acid sequence. But There are 20 amino acids. And we learned earlier that there are a codon, which codes for amino acids. There's 3 nucleotides per codon. And if you do the math, that means there are 64 possible codons. There's only 20 amino acids. In the genetic code, there is redundancy. So there's more than one way to call for a particular amino acid. And so when you look at the amino acid sequence of these two prestin proteins, it looks like the porpoise is related to the bat. But when you look at the DNA, you see that it's using different codons to summon those amino acids, and it's using codons that are almost identical to those in whales when the porpoise is doing it. So when you look at the DNA, it's obvious that this is a whale, where that was not obvious when you're just looking at the amino acid sequence. It's a really cool little thing that we found in the specificity of using DNA clocks instead of protein.
That's fascinating.
Yeah. Fountain of fascinating things you are, John.
Oh, why, thank you. We'll do the next slide here.
You're welcome.
So what types of molecules are best to use? Obviously, the more data we have, the better. So I would say, in a perfect world, use nuclear DNA and use mitochondrial DNA. But because there's, cost limitations and attention limitations, mitochondrial DNA has been really nice for doing these. And the reason for that is that your DNA is arranged into chromosomes. And a chromosome here on the left, I have some X-shaped chromosomes. That's kind of what most of our chromosomes look like when a cell is dividing in the nucleus. And a chromosome is just a big long chain of DNA wrapped around proteins. So in that X, there's actually 2 copies of the chromosome in there. And like I said earlier, half of your nuclear DNA comes from your mother, half comes from your father. So I've coded this pink and blue here. And when you are making sex cells, when you're making sperm cells or egg cells, those chromosomes come together and they swap chunks. So this is called genetic crossover. And the sperm and the eggs that you make are a really weird mix of your parents' DNA. And it gets really messy. This adds a whole new complication to the math that you have to put on top of your coalescent theory when you're trying to track evolution across the nuclear genome, across these genes, these molecules that do this genetic crossover. It's really frustrating. And it's not that bad, but it just adds another layer of complexity to the whole problem. Mitochondria don't do that. Their chromosomes are circular like this, and they just replicate. They make copies of themselves. They'll come together and do some crossover stuff to repair damage, but that's it. They're not shuffling genes. There's no shuffling going on there.